In this video, we will continue our discussion on this CAR T cell on solid tumor. In the previous video, we have discussed some of the results of the antigen, or that is anti-TN antigen against the TN antigen on different kinds of cells. So, some such as Jurkat cells that has this TN antigen, whereas the normal cells does not has TN antigens. However, in this video, what we will cover the results of this CAR T cells. We will cover the results of these CAR T cells on the primary cells, as the results of the primary cells are much more relevant, and also we will cover some of these CAR T cell results on some of the cell lines, or in other terms, in vivo studies results. Also, we will try to understand the in vivo study results. So let us start with the primary cell study results. So the CAR T cells studies are being carried out. CAR T cell studies are being carried out on these Jurkat cells, Jurkat 19 cos cells, cardiomyocytes, aerotytes, osteoblast cells, and some of the other primary cells. So they have tested these three kind of antigens, three kind of anti-antigens that is used in the CAR T cells. First one is the CD19. Sorry. First one is the CD19. Another one is the 5E5, that targets again anti-TN, that targets again TN antigen, and another one is this MC81370. So what they have find out is that this Jurkat E61 and in Jurkat Cosmo Cos MC, the lysis cells, or the effect of these cancer T cells in terms of they have measured the cancer T cells effect in terms of cell lysis. So they have seen that percentage cell lysis is increased in the Jurkat E61 cells and 19 comp cells, as the this TN antigen is TN antigen will be exposed on these kind of cells, as these are more they are more closely relevant to the cancer cells, whereas in the other primary cells these kind of antigens are not found, and therefore we do not see any signals for this CD19 and 5E5. However, contrastingly, we see the signals for this MC81370. So this might indicate that this MC81370 might have this cross reaction activity, and a that will produce a negative response or side effect whenever this MC81370 antigen is used in the CAR T cells in order to treat tumor, as it will consider the primary cells also as the tumor, and it will act against those primary cells also. Whereas for the CD19 and whereas for the CD19 and 5E5 are shown positive results only for this Jurkat E61 and Jurkat 19 cos MC, as they have the antigen, they have these properties that are similar to the cancer-like cells. Thereafter, they also did this dose-dependent study on effector to target ratio. That is, they provided they did this kind of studies are carried out in the cell lines, different cell lines, and against these CAR T cells that are injected at different concentration. And they have, in this case, they have measured the intracellular cytokine release, and intracellular cytokine release by the T cells. So in this case, the results shows that the intracellular cytokines release for this 5E5 B set is uh, almost almost below control level in this no target cell lines that does not contain anti-T and antigen, except the intracellular cytokines results are only visible for this GZ MB plus in the case of 5v5, whereas in all the non-target regions uh, does not show any significant increase in this intracellular cytokine production, whereas it shows intracellular cytokine production increase for all the other conditions such as K562 and Jurkat cells, MMP4 cells, it is the ovarian cancer cells, and this is the leukemia cancer cells. Jurkat cells is also related to the cancer cells. Whereas the another surprising thing that is found is that the CD19 does not show any signals, any intracellular cytokines signals for any of these cancers related cells. So provides the this provides us the understanding that the CD19 cells is less effective compared to these 5v5 cells. And entity is the non-transduced cells. It is act as a negative control. Later on, what they did is they analyze this lysis cells, percentage lysis cells at different effector ratio, and in this case also they have find out that this 5v5, or in other term they combine all this data, and also they use the different effector to target ratio to see the effect of this, to see the effect of these CAR T cells on the tumor antigens, and they have seen that 5v5 produced a more prominent response, as the percentage that in the lysis is more compared to the other competitors, such as other candidates such as the CD19BZ. So CD19BZ is not that much effective in detecting tumor cells compared to this 5v5BZ. Thereafter, they did the in vivo study on dose dependent manner, and in this case, they produced a they inject a xenograft of these tumor cells to the mouse, and after that, after 8 days, they introduce these CAR T cells to those mouse, and they see the dose dependent signals of the fluorescence, as the tumor cells is fluorescently labeled, and they can see that this is the negative control. This does not show any decrease in the fluorescence signals, and this is non-transduced cells does not show any decrease in the fluorescence signals, whereas the CD19 cells is also signals are also similar to this control signals, whereas for this 5v5 CAR T cells shows some slightly decrease in the flux signals or bioluminescence, and the increased concentration of these CAR T cells shows further decrease. Increased concentration of these CAR T cells shows further decrease in the uh, flux signals, and this is the overall figure combining combining all the combinations of in vivo studies. In this case, we can see that the 5v5 has the reduced signal, and with increase in concentration, the fluorescence signal or the tumors are further reduced. And thereafter, they detected this percentage survival capacity, and in this case, we can see that the patients that have this CD19 or non-transduced cells, or patients or not patients, just mice that has the CD19 or 5v5 CAR T cells are died earlier compared to the mice that has this 5v5 CAR T cells as the cancer treatment. And we can see that the with increasing dose of these CAR T cells, the we increase in dose in the CAR T cells, there is a intermediate increase there is an intermediate increase in the survival rate that can be seen here. So this is some of the data about this, how this CAR T cells that has this entity and antigen works on the solid tumor, and this finally forms as a basis in order to proceed these kind of CAR T cells for the clinical trial. Right now, these CAR T cells are in the clinical trial. If you need to understand more about these CAR T cells on solid tumors, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.